all the girls around the world, love and be loved. From Linda G and Lady T, baby.
I wanna talk about Linda good girl, yeah. 아직은 조금만 더 Linda 지치지만, yeah. I wanna talk about Linda good girl.
Hi everyone, how have you been? So, in a continuation of this chapter 3, which we have left off before midterm exam, so let's go over the energy aspect of this in the cell, which is mediated by two major organelle in the eukaryotic cell, respectively. One is mitochondria and the other one is chloroplast, which unfortunately we animals don't have. So overview of this cellular energy metabolism is, first off, we are going to take a look at the way through which we get to produce this the precious form of energy from the ATP, the universal. I compare this with the rechargeable battery. How to obtain this ATP, and so that we can utilize this in various many different ways. We all need this ATP as a how to use this ATP by breaking down this ATP into ADP, which is a catabolic reaction. So then energy will be released as a result. So then we take this energy to get some work done. So then how do we obtain this ATP? In order to utilize this, we have to be able to synthesize the ATP. From where? From this ADP. From this ADP we get to, that's why I compare this on a rechargeable battery. So how do we charge this ADP into ATP so that we can reuse this? Is a basic essence of what we are going to take a look at in the first part of this cellular energy metabolism. So which was summed up in this diagram. ATP, so it just keep being recycled. So in the meantime, so in this aspect, if you wanna make this ATP, oh here by the way, there is a typo. This should be ADP. Sorry. So from 2-phosphate diphosphate to, in order to build one more phosphate group containing ATP, so you need to attach this one phosphate group into, to do so, because that direction of the reaction is a anabolic reaction, so you have to provide the energy. How? By breaking down at the sacrifice of other molecule. By breaking down, then you get the energy and then provide this input. And then in turn, once you build up this ATP, now you utilize this by the similar way, by breaking down by means taking one phosphate away, then energy will be released. That's how you utilize this ATP energy, universal energy source. So to produce this ATP, although we can, in a cell, we can produce the ATP without having the mitochondria, the organelle involved, but without it, we cannot generate as much ATP as we'd like to have, as a, in particular, especially as a multicellular organism, which requires a huge, much higher amount of energy usage. So this mitochondria is that's another comparison. That's why I used to compare this mitochondria as a power plant in the cell. So the overall procedure, if we try to characterize this whole procedure, you need the food. So that this food is the one that the source of this energy that we are going to take in order to build the ATP. So from food molecules, so we break down this food molecule, which is a catabolic reaction, so that energy will be released, and then we use that energy, convert the energy into the building procedure of the ATP. So this is a whole the food. So we can utilize so many different diverse source of the food, but one very good representative of such a food is glucose, because it is very convenient. Such one good example of such high energy containing organic molecule. Once again, reminds you that only organic molecule for us, only organic molecule can serve as a food for us. So because we are not autotroph, so we lack the ability of converting the inorganic molecule into the organic molecule. So we have to start from the organic molecule, and the glucose is one of the simplest and very good, efficient such organic molecule that can serve as a food. So by breaking down this glucose molecule, by the way, this glucose molecule contains 6 carbons and 12 hydrogen and then 6 oxygen. This formula, chemical formula, of course, none of us have to memorize this, but just to put it this way, so having this many carbon molecule which are all connected together, one particular chemical bond called the covalent bond, so that makes this glucose molecule one such a very good example of a good candidate, representative candidate of the food, that we have a plenty of area that we can attack and break down their chemical bond. So it is the type of reaction takes place, which actually, to be more accurate, it doesn't start from mitochondria, but it culminates in the mitochondria. The overall procedure is a 3-step procedure, 2 of which process are taking place inside of the mitochondria. So overall, to characterize this whole entire procedure, we can say that as a catabolic breakdown of glucose through the name of this whole entire procedure is called the aerobic respiration, because it requires the presence of oxygen. So the aerobic respiration is what this entire procedure is called for. So then the end result, what happens is in the presence of this oxygen, the glucose molecule can be completely oxidized. So we are going to take a look at to distinguish the one type of a chemical reaction called oxidation, and the opposite side of the chemical reaction is called the reduction in a minute. So just follow with me. So the entire process can be called as an oxidation of glucose into carbon dioxide. CO2 will be released, that as we know. And so we need oxygen. So oxygen has to be provided in this procedure, and carbon dioxide will be released at some time point in the procedure. So this is glucose is oxidized, and in turn the oxygen molecule that enters this reaction will be reduced into water. So here's the bottom line of the one particular type of chemical reaction called oxidation and reduction. This chemical reaction always occur at the same time, and this is opposite. So if oxidation reaction occurs, then it has to be accompanied with the reduction reaction, which goes to the other opposite direction. It's always, so it is a two-way, always a two-way street. So if one molecule is oxidized, okay, let's say chemical A and B are the participating in this chemical reaction. So let's say A is oxidized. How do we actually usually view oxidation reaction? One very simplistic apparent way is if this one combines with oxygen, then it is being oxidized. Okay. Then if this one is oxidized, in turn, the another molecule participating in the same reaction is going to be in return is reduced, which means it becomes free from oxygen, is what we can probably understand the oxidation and reduction. So to sum up, the simple way of explaining or understanding oxidation reaction is if you obtain oxygen, the rust ion is oxidized. The ion becomes reddish, something when it combines with, through this oxidation reaction combines with oxygen, then we actually take notice that ion becomes turned into something a little bit different, the status, because it is oxidized, it combined with oxygen. Okay. So one apparent way of witnessing this oxidation reaction is if one molecule combines with oxygen. That's an oxidation. So oxidation, combine with oxygen. Okay. Then the reduction reaction also has to take place at the same time. If one molecule is, one particular type of chemical is oxidized and the other one is in turn is being reduced. Reduction means away from oxygen. If you run away from oxygen somehow, so you are free, then you are reduced. But things don't always show this very straightforward landscape, so to speak. Another way of looking at, so in some cases actually you cannot really trace directly the movement of oxygen. So one chemical, apparently without being involved in directly the combined with or being away from oxygen, still the chemical can either be oxidized or reduced. In that case, you have to follow the electrons. So another way of, so is an or. If you get to combine with oxygen, you are apparently oxidized. Or if you somehow lose your electron is usually described as a E and minus, then you are also oxidized. In other words, as a chemical then being reduced, the opposite direction of the oxidation, even if you don't get away from oxygen apparently, but if you somehow obtain electron, then you are called reduced. So either way, so either the movement of oxygen or the obtaining or losing the electron can both describe the status of a chemical in terms of this oxidation and reduction reaction. Why do I take pains to this stupid concept to you in the understanding introductory course of the life science? Because this entire procedure that we are going to take a look at is actually nothing but the oxidation reaction. So from the perspective of this glucose molecule, that we are going to break this down so that we can extract the energy from glucose molecule. So what is the fate of this glucose in the overall, so to speak, aerobic respiration that we are going to take a look at now? Is actually glucose is oxidized. How do you know? Well, glucose itself is somehow a little bit difficult to trace that, oh, glucose is finally combined with oxygen. Sometimes it may not be such like straightforward as you like to see. However, it is clear that glucose is oxidized because glucose, the electrons originally present in the glucose molecule, is being stripped away. It is not all at once, but it is very cleverly, one by one, slowly, just like instead of going down from the cliff at once, it is going down like in a stepwise way, so that it slowly releases its electrons. This is so. Actually, this is a very clever and energy efficient procedure. So that if we do this, then a lot of those energy originally present in the glucose molecule will become it's just such a waste, so that we cannot take advantage of. But by doing this stepwise conversions, the oxidation reaction, the stripping away remaining electrons, then we can somehow maximize the current level of the technology in the cell, maximize this energy utilization of glucose molecule. So here in this figure, the A and B two molecules became A plus and B minus, which means one has lost electron. Then if you lose the electron, then you become a positively charged molecule. And another one in turn obtained the electron, which originally was there in A molecule, but this electron was transferred into B, so that B is now the negatively charged molecule. So in this case, then which one is reduced and which one is oxidized is by virtue of this electron transfer, we can determine the A was the one who got oxidized because it has lost the electron, and B has obtained the electrons, so B was reduced. Okay, fine. In terms of energy, so that you might actually be curious. I mean, in this reaction, which one is a catabolic and which one is an anabolic reaction? So which one gain the energy and which one lose the energy? This figure kindly is providing the clue, so that it's in color. It's orange, and which one is orange? This one is yellow and this one is orange. Let's determine. Okay. So this yellow color indicate the lower energy status, and this orange color indicating a high energy status. So at the end of in the beginning, there seemed to be virtually no difference in the energy level in between A and B molecule. But then at the end of the reaction, one is oxidized and the other ones got reduced. But if you look at this color, A it's become lower energy, which means if you had a certain amount of energy, but now your energy state has become lower, that means you have given off the energy. That means it's a typical characteristics of what? The catabolic reaction. So this being oxidized means equal to catabolic reaction, which means you are going to release the energy. So that's why we are trying to oxidize this glucose so that we can take off the energy. Then in return, the one is being reduced will take those energy released. So as you can check this color, so B, after picking up the electron, now it's become a higher energy status. So which means it's being like it's experienced the anabolic reaction. So it's got obtained high energy. So if we plug this whole the scenario into the case of this glucose oxidation of this aerobic respiration, glucose is being oxidized, so it will release the energy. Who is going to be reduced? If glucose is oxidized, then somebody else has to be in return go through the opposite path of reduction. So who is the one who gets to be reduced? Oxygen itself. So now oxygen, at the end of this whole reaction, oxygen is reduced. Once oxygen is reduced, which means getting the electron. Remember, oxygen is one of the greatest molecule for electron. Oxygen is by far in the living system, inside of the living system, probably by far the oxygen is the most electron hungry. So it will try to take, it will not think twice. Any opportunity so that he or she, oxygen, can take an electron from somebody, or doesn't think twice, and then it will take it. And it has such power. Okay? Will and power is all there in the oxygen molecule. That's why we have to be careful about being contact with oxygen. So the reactive oxygen, mind you, is a very dangerous molecule, reactive oxygen, which is the culprit of the aging procedure, and a lot of damages can also be done by the activity of those such higher activity oxygen is what we define as a reactive oxygen. So oxygen is essential to us, but at the same time oxygen is inflicting a lot of enough damage to our presence, existence too. So anyway, so oxygen is reduced into water after getting the electrons so that it can pick up hydrogen. Usually the flow of travel of the electrons, the reason why oxygen can become water by picking up hydrogen molecule, and then now we declare this one oxygen has been reduced, is because usually electrons travels in the form of hydrogen, the proton itself. So that's why. So this is overview of such procedure in the cell, which here imaginary cell, a single cell, and I just only drew one mitochondrion. Of course, in the cell there are many, even hundreds, or even 1,000 mitochondria molecule doing the same things, but for the simplicity I just only leave one such mitochondrion molecule and skipping everything else in the cell to simplify this and to illustrate this whole entire procedure. So as you can see here, 1, 2, 3 different labels representing 3 different stages of the entire reaction, which begins with this glucose molecule having 6 carbons and 6 oxygen molecule. Once it enters the cell, then the procedure begins. So we are going to take a look at this whole procedure now. So the first step here, which we named as a glycolysis, because here what is basically the glycolysis is we can describe as partial breakdown of glucose. So as you can see in this figure, the glucose molecule is cut in half, like the French baguette. Long French baguette is too long to eat at once in your mouth, to fit into your mouth. So you have to first you try to cut it into half, into smaller piece. Is what exactly happening in this first stage of this procedure. So that's why we can call this procedure of what glycolysis or whatever, this partial breakdown of glucose. And cytoplasm means the place of this. Where does it happen? It takes place in the cytoplasm. Cytoplasm means, once you, for those who Munhwa students, to remind you, cytoplasm is the whole area inside of the cell except the nucleus is in the cytoplasm. And what do we get? So this is kind of the bullet point summary of the whole procedure. To simplify, although it is extremely complicated chain of reactions, I will try to make this whole thing look easy as much as possible. And it doesn't really compromise the understanding level of it. Just because I just simplified this thing doesn't really compromise any level of understanding. That's what I personally believe. So the gain is at the end of this procedure. If somehow if we stop this entire procedure, instead of going all the way down to the very end of this so-called aerobic respiration, but if we quit over here, hey, I don't like this, I will just quit. Still, what you get is this small amount of ATP, very small. Actually, per one glucose molecule, actually you get to have a 2 ATP molecule, which is really is not enough. But sometimes you have to, if you don't have any alternative, you would have to live on this very small petty amount of ATP. That's what many of those organisms actually have to live by. But anyway, and another thing that we have to take note is up until at this point, even though oxygen is like love, so you cannot live without oxygen as the multicellular organism. But up until this stage, actually, there is no requirement for an oxygen. So if you can somehow stop up until this and then somehow survive, then you can still, you don't really have to breathe in to survive. So that's the thing. Now, let's move on to the second stage of this respiration, aerobic respiration. Number 2, here, as you can notice, now carbon dioxide, the CO2, is being released at the end of this procedure. And the name is, several different versions of the names are available for this particular procedure. One such version is a TCA cycle, tricarboxylic acid cycle, standing for, and also another name is a Krebs cycle. Krebs, the scientist, the name of the scientist who figured this thing out. By the way, I forgot to mention the first stage, which is glycolysis, which appeared to be deceivingly simple procedure. Simply, what would it take? You just simply cut the glucose molecule into half. It's a very simple procedure. However, to accomplish that, seemingly very simple procedure. What it takes is 10 different chemical reactions catalyzed by 10 different enzymes. Without it, you cannot make those procedures go through. So uh, that is actually in actually it is a very complex chemical reaction, which without the involvement of the enzyme, there is no way you can accomplish it within the reasonable amount of the time. Even though glycolysis per se is apparently a catabolic reaction. What's characteristic of a catabolic reaction? It is a spontaneous reaction. So it will happen without any intervention. If you leave them, and then it will happen. Just like hydrolysis of ATP into ADP is a catabolic reaction and is a spontaneous reaction, so that it will happen spontaneously. However, so glucose molecule will become whatever into two halves through the glycolysis. This glycolysis, spelling, whatever. Actually, this name of this chemical, the hefty glucose, is a pyruvic acid. We call it pyruvate. However, whatever the name is not important. This is also catabolic reaction, which is a spontaneous in theory. However, if you simply leave this glucose molecule to become pyruvate into two these things, hey, this is a spontaneous reaction. It will happen no matter what. It will take about 5,000 years to become one glucose molecule to become eventually to pyruvate. Nobody has such a patience to wait for as long as 5,000 years after you have your snack or lunch or whatever, as a sandwich or pop or anything. So that's why you need the involvement of the different the enzymes, because those enzymes will dramatically speed up this whole reaction, so that it now in the presence of those enzymes, it can be accomplished within like tens of seconds, 10 milliseconds. I don't know, I didn't really check exact amount of time that it takes to convert this glucose into pyruvate. So anyway, that's the importance of the enzyme. Even if there was a spontaneous reaction or a catabolic reaction, that is the reason why we need such the enzymes involvement in chemical reaction in the cell. So getting back to this second stage of respiration, the TCA cycle or Krebs cycle, whatever, is also another very complicated chemical reaction, and also the thing which is slightly different from the first stage, which is the glycolysis, is this chemical reaction is arranged in circle. So like this. So it is it seems like it's a revolving door. So the chemicals entering this cycle is keep being cycled through all these chemical reactions instead of the linear fashion, one starting point and an end point. But this is like keep circling around, and then one reagent is entering this cycle at certain point, and as a result, at the end, the product is being released out of, and then just keep the continuation of this cycling fashion of the chemical reaction. is the characteristic of this such the cyclic reaction. So once again, with so much of this babbling aside, this second stage of this whatever the TCA cycle or Krebs cycle, whichever you call it, is arranged in cyclic fashion. The whole several different chemical reactions are arranged in cycle, and to characterize this cycle, what we can say is now it is a complete oxidation of glucose, as you can see over here, carbon dioxide. So this glucose is now become the entity of some in from for the sake of this carbon molecule. This is the maximum level of oxidation for each carbon. So in other words, this glucose molecule originally having 6 carbons. Now, once they go through the end of this second stage of this cyclic reaction, 6 carbon dioxide molecule will be released. So that carbon dioxide is the one that we breathe out, the gas that we discharge. And this mitochondrial matrix means where it takes place. The mitochondria itself, as we all know by now, hopefully, consists of 2 different layers of a membrane, the outer membrane, and the inner membrane has some interesting shape of inwardly folded. This structure is a typical architecture of the mitochondria. And so this outer membrane, OM, and inner membrane, IM, and so entire internal space, how do we call this internal space? Mitoplasm? No. That's what we call as a matrix. Mitochondrial matrix, the internal space. So this is where the cycle, the TCL cycle is taking place. And the result, what do we get? Once again, still you have a very small amount of ATP at the end of as a result of this reaction. But overall, that's not really enough for us entire big body to survive and rely on. And more importantly, actually, what's actually the importance of the second stage, the TCA cycle, is this so-called the factors we call as a core factors. They are acting as electron carriers. Remember, this entire procedure is nothing but the oxidation reaction, which is something that we can view as the transfer of electron from glucose into somebody else. So being the electrons originally present from glucose is being stripped away. And who is going to take the role of the electron messenger, electron carrier? These guys we call as cofactors for electron transport, some of them. So namely, we don't have to memorize, but their names are NADH or FADH, NADH2, FADH2, like so. These, once they are oxidized, and then this hydrogen will be taken away, and once this reduced, then it will be able to pick up this hydrogen, because hydrogen is the one actually that comes and goes together with the electrons. So these two are NADH and FADH are the two such electron transport carriers. We call them as cofactors for some reason that I don't have to go over in this class. So this is the one of the most significance of this TCA cycle, because at the end of the day, which is the final stage of this aerobic respiration, the role of these electron transporters very significant, because they are the one who actually finally release their electrons originally that came originally from glucose. Okay, here they are. These are the shipment from glucose, so that take as many as you want. So then those electrons are being processed, and then through which, during that procedure, very big amounts of ATPs are produced, and that's the essence of this whole entire procedure. So, and another important thing of this second stage is now still there's no oxygen is actually required. Even though you see, hey, what about this oxygen? Where did it come? Because carbon is now completely oxidized. Look at this carbon dioxide. For the sake of the carbon, there is no further room for this carbon molecule to pick up the oxygen. So literally and actually graphically, you can see, oh, this is completely oxidized. But then where is the source of this oxygen if it didn't come from outside of source? Actually, this oxygen is the one internally this glucose molecule had. So this oxygen is not the one that we breathe in from the air. So in other words, theoretically speaking, up until the second stage, you can do this thing without any breathing in, without any supply of external oxygen is the thing. Although in practice, the second stage does not even happen in the absence of oxygen. If the cell senses, oh, there's no oxygen, and they know, oh, there's no point of getting into this whole messy business of a second stage, so let's call it a day, and they stop whole procedure at the end of the first stage, which is glycolysis, is what's happening in reality. But in principle and in theory, without even oxygen, this second stage of a TCA cycle can even be processed and can then be completed. This is a little more realistic the representation of this the second stage, which is the TCA or Krebs cycle. So many different intermediate chemical reaction, and we get to see NADH and FADH2, these two forms of electron carriers, cofactors, are generated throughout this, and both of them will be fed into the final stage of this respiration that we are going to take a look at now. As you can see in this figure, try to emphasize that the amount of ATP generated from this stage is huge, so you get to see so many ATP bursts in this figure. So the name of the final stage is electron transport chain. Once again, names are not at all important though. So this procedure, what happens in this procedure is the oxidation of the electron transport cofactors, those guys. Let's go back. If the second stage, the electron cofactors, these two, are the one actually generated as a result of the reduction. They are the one actually the reduced product. Look at this hydrogen. If you see this hydrogen, then you kind of sense that, oh, they are the one actually now at this point. For now, they are reduced one. And always this oxidation reduction reaction is always a two-way street. Somebody like this pyruvate, which is the half of the glucose molecule, is eventually oxidized so that they lost their electrons to these guys to become this reduced form. Now they will enter the final stage, and then their fates are reversed completely. their electrons will be taken by the stronger guys. Okay, stronger bullies. Hey, you guys have electrons, give me that. And then they will lose, helplessly lose their electrons, and then will return to their home to pick up once again their electrons. And these are the ones that is keep being cycled. So here, oxidation of the electron transport cofactors is the principal reaction to what's happening. And if those cofactors, NADH and FADH2s, are oxidized, then somebody else in turn will be reduced. In the middle, actually, there are some other those enzyme complex who initially takes the electrons from those cofactors, but eventually, at the end of the day, eventually all these the electrons will go to the guy, who the oxygen. So oxygen is the final ultimate electron acceptor of this the business. And where does it happen? Inner membrane of mitochondria. So if the second stage, which is the cycle, Krebs or TCA cycle, took place in the middle, the internal space, which we call the matrix, then now the final stage is actually, as you can see, the inner membrane, along with this inner membrane of this mitochondria lies several, actually 3 different enzyme complexes. 1, 2, 3. 3 these enzyme complexes who are involved in this electron exchange business. So like the first guy of this enzyme complex take those electrons from the carriers, cofactor carriers, NADH and FADH, and the next guy, the second enzyme complex, will take those electrons from this guy, and then the other guy, the third guy, or another enzyme complex will take that electrons again, and then those electrons will be given up to finally to the oxygen. So that's what we recognize. This oxygen is being reduced after taking all those electrons and then to become water. So here, entire procedure of the electron transport chain, the final stage, the importance or significance is the amount of ATP being produced is that there's no comparison between this final stage and the other two. So huge amount of ATP is being produced. In the general biology, sometimes, a lot of times actually, it gives you specific numbers, something like a 36 ATP per glucose molecule, etc. But who cares? How many? It's a lot. That's just enough. A lot of ATP it can be generated through once you can go through, once the glucose molecule can finally go through up until the final stage of this electron transport chain, then now the reward is a huge amount of ATP. But how is actually more fascinating things that you might want to actually take a look at it. Instead of sort of breaking down and then get the energy or something, but this is some kind of so to speak, revolutionized way of generating ATP by using some kind of the concentration build up in a very small space. So here the keyword, the mechanism of this ATP synthesis in the electron transport chain is 2 keywords: hydrogen ion. The hydrogen ion and the concentration gradient. So this hydrogen ion accumulate in a very small space, where very small space created between the outer and inner membrane space. And the hydrogen ion gets to become so crowded over there, and it builds up a huge potential. I want to get out. I want to get out. If you blow in the air in the balloon, then eventually it will try to either go back or go burst. Okay, so either way, you have to provide the way out, and that is a strong actually power with which you can generate the synthesis of ATP from ADP. So this concentration gradient means at one end of the space you have a huge concentration, high concentration, and the other end of the space, if you have a lower concentration, then by nature this molecule will try to move from the concentrated area to the lower, the concentrated area. That's the principle of, so to speak, the diffusion, isn't it? So the same principle of this whole entire universe is being utilized in this step. So using this power, the concentration gradient of a hydrogen ion from one very small place to the other way out, then now you can synthesize ATP from ADP. And that's, so if you zoom in this small space, what happens is, so this guy number 1, 2, 3, these 3 electron transport the chain, the enzyme complexes will take the electrons in the form of this that present in the hydrogen. And actually it can separate these electrons from hydrogen now. What happens is they transfer these electrons from this guy to the next guy and the other guy, and then eventually provide to the oxygen so that the oxygen can become water. In doing so, electron will travel. So this is the oxidation reduction reaction. Why? Because if this one is oxidized and that one is reduced, and then in turn if this one gives up the electron so that become oxidized, then this guy is being reduced. So that's why entire whole procedure of the sequence can be viewed as oxidation reduction reaction, eventually culminated in the reduction of oxygen into water. So electron will flow, but at the same time electron originally present in the form of hydrogen, but now it's separated. So electrons flow, but at the same time the hydrogen, which now devoid of electrons, that was a proton. If a hydrogen element, as an element, hydrogen being rid of the electron, now it is an ion, proton, hydrogen ion. This proton equals hydrogen ion. This proton is pumped into the internal space between outer and inner membrane. Why? Because these 3 guys very similar to the proton pump that we have seen previously in the lysosome, membrane of the lysosome, as using the ATP as an energy source and actively pump the proton ion into the small space. That's what this proton pump present on the lysosomal membrane does. And similarly, our stomach membrane, the similar proton pump present, and then pump the proton ion into the small, is not small, the gut, the stomach space, so that's why our internal environment of the stomach is highly acidic. However, so these guys, the the enzyme complex of this electron transport channel can do the same thing. So they can act as a proton pump. However, one distinguishable characteristic of these proton pump on the inner mitochondrial membrane is do not need ATP as an energy source. So even without such ATP energy, the hydrolysis, these can act as a proton pump so that increase the concentration of the hydrogen ion within this small space. Now what? If this hydrogen ion concentration build up, then they will try to escape this small space because due to this huge differences between concentration. That's what we call as a concentration gradient. However, the only way out is this space provided. Yeah, there is a one way out. However, this will provide this, the space, the tunnel out. However, once the hydrogen, if hydrogen ion is trying to pass through, it has to pay, so to speak, it has to pay the fee. That energy will be the moving out, such potential energy will be converted into the other form of energy with which this ADP can be converted into ATP. That's how you generate this energy, ATP. The actual thing what's happening in this is, so to speak, this is called ATP synthase enzyme complex. ATP synthase or synthase. Because by doing so it generates this ATP from ADP. So at bottom, actually, this bottom of this enzyme complex, there are very small rooms in which each room can accommodate each single molecule of a proton hydrogen ion, and its shape, its architecture is very much similar to the pistol, revolver pistol of the bullet case, and then it can actually rotate and then shoot one hydrogen at a time, and that shooting motion can somehow provide such mechanical energy, can be converted into this type of the synthetic energy from ADP to ATP, to put it kind of a little more understandable way, or maybe not. So this is the structural model was present on the cell membrane, the mitochondrial membrane. There are three different enzyme complexes plus the ATP synthase enzyme complex too. So that's the whole entire summary of this aerobic respiration. And like I said, this entire aerobic respiration, the one very important premise, the condition is it can take place only when the oxygen is present. But what if then oxygen is not available? A lot of different kind of a small sub-environment as well as some overall environment organism can be subjected to an oxygen less environment, or more commonly the availability of oxygen is not enough, then what are you going to do under such circumstances? The thing is, these three different stages of aerobic respiration, which can be separated into each stages can be separated, and actually, if we consider our entire body, actually, notably, there are only two very distinguishable two area where the demand for oxygen is so enormous. So that the brain and heart are those two notable area where you have to have huge amount of ATP being provided. For these two area, brain and heart, if you cannot provide such enough energy in the form of ATP, then they, in a matter of minutes, their cells, the tissues comprising the cells comprising such tissues and organs will die. So that's why you as an entire organism die when you are suffocated. If you don't have any oxygen, you die because of this. Brain and heart cease to function, which are most of which are very, very critical for your function, of course. However, speaking of the rest of the area, they can actually manage it even without the enough supply of oxygen, because they can survive without going through at the end of this the electron transport chain, but simply by only doing the glycolysis. So from that angle, we are going to take a look at this alternative metabolism. We call it the fermentation. So this fermentation is the kind of a metabolism, and like alternative route of this glycolysis when there is no oxygen available. We all know that actually some cells can survive without an oxygen. Some it's because they can carry out this glucose catabolism, namely it can go through this glycolysis on the anaerobic condition, because glycolysis per se doesn't require any the oxygen supply. Also, the second stage, which is a Krebs cycle, can also be accomplished without the oxygen, but like I said, cells sense the lack of oxygen, then they don't even bother to start the second stage of this the aerobic respiration. So that's we don't have to consider that stage. So this fermentation, instead of stopping at the glycolysis, actually what happens in this fermentation, this end product of glycolysis is further converted into something else, like being metabolized. So, for example, in yeast, which is a very famous metabolic product of this fermentation, as we know, the alcohol. So in yeast, they go through this so-called alcohol fermentation. However, in our muscles, instead of producing alcohol, they have another alternative product of this glycolysis. Is a lactic acid. Lactic acid is also called a lactate. In higher animals, like it is apparent that the reason, instead of unlike yeast, instead of producing alcohol, the lactic acid is being produced is apparent, because in our muscle, if we somehow in an environment where the oxygen is scarce, oxygen is wanted, then if we get to produce alcohol, then it will be disastrous. So it is actually very wise things to produce lactic acid under oxygen lacking environment instead of alcohol for us. But anyway, the fermentation reaction, the end result of this fermentation reaction is, if we try to look at, it seems like a waste of energy, because to further convert this end product of a glycolysis, which was the pyruvic acid, so through fermentation, either it becomes alcohol or lactate. Either way, to do so, you actually have to spend your energy, and also you have to the final yield of the ATP is reduced into half. So you might wonder, actually, why are we doing this? This is totally waste. But the fermentation is extremely important in the anaerobic condition, which is like the air in an environment we don't have any oxygen supply enough. Why? Because in order to continue the glycolysis reaction, you have to secure this raw material to start with. Actually, the raw material to start with, among many other things, the one of the most critical such raw material to get this glycolysis going is NADH. No, it's not NADH, but NAD, the oxidized form of this NAD, which is NAD plus. Without this, you cannot start this glycolysis. Normally, when the oxygen is plenty in our cell, the most of this source, where do all this most of the NAD plus, the oxidized form of those electron cofactor carrier, come from? The mitochondria. When the NADH, they all lose their electrons to the enzyme complex of the electron transport chain, then they will become this. So we don't have to worry about the supply of this NAD plus to get this glycolysis started. However, now you got a problem in that you cannot do this, the final stage of the aerobic respiration, because you don't have any oxygen. So the supply of this NAD plus is cut off. So how are you going to get this NAD plus supply? At the expense of these things. Because I didn't tell you, but at the end of this glycolysis, you actually have very small amount of this NADH produced. So you are going to use this NADH. That's what we view the fermentation reaction as a waste of energy, because if we did not use this NADH in the fermentation, this NADH will be a very good source of generating huge amount of ATP at the end of the aerobic respiration. But that's a moot point where you don't have any oxygen. So that material, NAD plus, becomes, like I said, you normally will be available plenty when the electron transport chain is incomplete, but on the anaerobic condition, you don't have such thing available. So this diagram will summarize this. So now what you do? This pyruvic acid, instead of getting into anaerobic condition where no oxygen is available, you have to take different routes in this fermentation. So what happens during fermentation? You have to spend this NADH to generate NAD plus. Why? Because this NAD plus is the one needed at the start of this glycolysis. That's the reason. That's the reason why organisms go through this fermentation. regardless of who you are, whether you are an yeast or you are a part of a multicellular body like your leg muscles. When oxygen is not available or not enough, then you immediately turn on this alternative metabolic pathway. Why? So that you can survive. Under this condition, where total machinery of a huge ATP generating machinery is not working, and the alternative, in order to survive, at least you have to live on very small amount of ATP that glycolysis alone can provide. So then your major concern is, how do we get this glycolysis keep going? And the only way, under the circumstances, the only way you can assure the continuation of this glycolysis, even though you don't like this glycolysis because it gives you only very small amount of ATP, but without it you cannot survive. So that you have to convert this entire procedure into this at the expense of this precious NADH. Otherwise, it would have been funneled into this mitochondria, and then you will get a huge reward. But in this particular circumstances, so that's such a huge silly luxury. So you don't do that. That's the essence of a fermentation. So in our cells, lactate is being produced like in two-step conversion. In yeast cell, they have this, go through this route to eventually produce in ethanol. For many different organisms, like many microorganisms, some can actually do this fermentation if they are needed. Those are called the facultative anaerobes. So when there is no oxygen available, then I can turn on this fermentation route so that I can survive. But some other organisms, absolutely they hate the oxygen, so they are called obligate anaerobes. So they require the environment, a very specific micro environment where oxygen is cut off, no oxygen is available. So different lifestyles of different organisms. So now, next in the following session, we are going to finish the other type of metabolism, which is a very good example of anabolic reaction. Up until now, what we have looked at is the very good example of catabolic reactions. That actually, in general biology, takes up the entire independent chapter worth of the material. So then we just simply concisely summarized in the 1 hour lecture, and then going to move on to the other different types of reaction. Photosynthesis, unfortunately, we do not have any ability